Alright, so we're going to pick up Chapter 4, Part B here. And so let me just remind you, we're looking at prokaryotic cells. And we've started on the outside and we're working our way in. So in the last part of Chapter 4, Video A, we looked at the appendages that would be attached on the outside. And so what we're going to look at in this section are the outer coverings of a prokaryotic cell. And so we call this area the cell envelope. And so if you think about just a general envelope that we use, it's something that's useful as an external coating to contain some type of content. Like, for example, if you were going to mail a bill, you would not just stick cash in the mailbox, right? You put cash or check in an envelope, you seal it, put it in the mailbox, and then it's protected. So the cell envelope is that external covering. It surrounds the inner contents of the cell, and it serves as a protective mechanism. Of course, there's some other functions that we'll talk about. And so for a cell envelope, it's typically composed of two layers: a cell wall and a cell membrane. So the cell wall being the outermost of the two, and the cell membrane being the innermost of the two. Now, as far as bacteria are concerned, you're going to hear us talk a lot this semester about gram-positive bacteria and gram-negative bacteria. And so what we use, these are two major categories of classification when we're trying to identify a bacteria, because some bacteria are gram-positive, some are gram-negative. And so what we look at with these is the main difference you're going to see has to do with their cell envelope. So if we look at gram-positive bacteria, you're typically going to notice a very thick cell wall made of peptidoglycan, and then the cell membrane just inside of it. So if we look at a picture, just kind of coming over here, of gram-positive, here's the two-layer envelope. So very thick cell wall, and then the cell membrane just inside of it. But if we look at gram-negative bacteria, we actually see a third layer. So you have what we call an outer cell membrane, a thin cell wall, and then the regular cell membrane just inside of it. So where gram-positive bacteria only have a two-layered cell envelope, gram-negative have three. So again, if we look at a picture, here's an outer membrane, there's the thin cell wall, and then here's the inner cell membrane. So that's just kind of a basic difference that we see with those. So what we want to do is take a moment and focus on these basic layers. So we want to look at the cell membrane, and then we'll look at the cell wall, and then we'll look at the cell membrane. Okay, so the first question is, why would a prokaryotic cell need a cell wall? Well, think about a wall. What does a wall do? Why in history did cities build walls around them? Well, one, it made an outer boundary of the city, right? So it allowed you to know where this city began and ends, right? So it kind of gives you an outer boundary, maintains shape, but it also for protection. Now, for cells, the cell wall, it provides shape, right? But it also provides protection in that it prevents lysis. When we talk about that word lysis, it just simply means to rupture, right? So what a cell wall essentially does is it keeps the cell from taking on too many fluids that would cause the cell to burst or to rupture. Now, as far as the structure of the cell wall, basic structure is peptidoglycan. So if we look at that, break that down, we know that peptid typically refers to like proteins, amino acids. Glycan obviously refers to carbohydrate or sugars. So when you combine those proteins and sugars together, you make what we call peptidoglycan, very strong material that composes these cell walls. So you can see a little bit more about how these are bound together. They're bound together in kind of a lattice work that makes them very, very strong structures. Now, one of the things that we want to notice, and we kind of have already mentioned this, has to do with the difference in cell walls from gram-positive versus gram-negative. So if we look at gram-positive, we already know from previous slide that the cell wall in a gram-positive is very thick. So you have a lot of peptidoglycan that's in here making up this really, really thick layer. But peptidoglycan is not the only thing that's found in the cell wall of gram-positive bacteria. You'll also find some extra acids. They're called teichoic acid and lipoteichoic acid. And so with the addition of these extra acids, it causes the cell wall in prokaryotics, in gram-positive prokaryotes to enlarge. So it makes it much larger than you see in other types of cells. And then of course you'll find this, what they call periplasmic space, in between it and the cell membrane. So that's what we see with gram-positive. When we look at gram-negative, again, we have already mentioned, here's that really thin cell wall. It also contains peptidoglycan. But in addition, you're going to find here some lipopolysaccharides that form this outer membrane here. We typically just refer to these as LPSs, but lipopolysaccharides, which make this third extra layer here. So gram-positive have the really thick cell wall, gram-negative have the very thin one. Now, before we go any further into the cell envelope, we want to take a minute to compare some reactions that we see. Now, you're going to learn in lab a technique or a procedure called Gram staining. Now, Gram staining, it's named after Gram. That was who actually developed the procedure, but the purpose of it is for identification. And so what we want to look at just briefly, and I'm not going to walk you through how to actually do a Gram stain, we'll do that in lab, but Gram staining, it's a differential stain. So it's a way of staining bacteria, dyeing bacteria, in order to distinguish a gram-positive cell wall from a gram-negative cell wall. So really quickly, just to kind of give you colors, a gram-positive bacteria will always stain purple. And the reason they stain purple is they retain the crystal violet, which is a very deep purple dye. It's the primary stain involved in gram staining. Gram-negative bacteria actually lose the crystal violet, and they take on what we call a counter stain, which is known as saffron, and gives them kind of a reddish or pinkish color. So before we go any further, just note gram-positive is going to stain purple, gram-negative is going to stain reddish pink. And this stain is so very important in bacterial identification. So we're going to be using this a lot this semester. So why do the bacteria stain different colors? Well, in the first step of gram staining, you take crystal violet, which is a very strong purple dye, and you pour it over the bacteria. And so what automatically happens is that purple dye, it goes into the cell walls and it makes them retain a purple color. So if you look at whether gram-positive or gram-negative bacteria, after step 1, they both appear purple. You'll notice this is what they're showing here. They're both going to appear purple because the purple dye is found inside of that cell wall. Now, the second step in gram staining is you pour iodine over the two bacteria, over the bacteria. Now, if they are gram-positive, they stay purple. Gram-negative stays purple, because what happens is the iodine actually takes all that purple dye in gram-positive cell walls and it traps it. Now remember, the cell wall in gram-positive bacteria are very thick, so there's a lot of room to trap those crystals, and so continuing their purple color. Iodine has literally no effect on gram-negative bacteria because remember, it's a very thin cell wall, so there's not really any place to trap anything, so it stays purple just because the iodine has no effect on it. So then we go to step 3. Step 3, we pour decolorizer over the bacteria. Now, decolorizer is a combination of alcohol and acetone, and it's called a decolorizer because the job of it is to strip color. But you'll notice that once we add decolorizer to gram-positive bacteria, it stays purple. Why is that? Well, remember, when we poured the iodine over the gram-positive bacteria, it trapped all those crystals. And so once they've become trapped, decolorizer does not strip the color out. So after step 3, gram-positive bacteria are still purple. Gram-negative bacteria, though, here you'll notice they are clear after this step. Why? Well, remember, iodine had no effect on those crystals, and so once you pour decolorizer over it, it weakens the cell wall, and all those color pigments are stripped away. So after step 3, gram-negative bacteria appear clear. So then we go to step 4, which is where we add the counter stain. The counter stain is called safranin. It's a reddish pinkish dye. And so when we pour safranin over gram-positive bacteria, the red dye has no effect. Why? Well, because purple has already been trapped in there and there's no room for the red or the pink. When you look over here, though, remember all the purple dye had been stripped away. And so once that red safranin is added to a gram-negative bacteria, it's going to retain the red dye, leaving the reddish or pinkish appearance. So you want to make sure, not that you know how to do a gram stain, because we'll learn that in lab, but the color changes that occur or the reactions that occur at each step of a gram stain. Now, the last couple things we'll look at in this section is some exceptions to the rule. So one thing we want to be mindful of is that not all bacteria have that ideal cell wall situation. There are some that have what we call non-typical cell walls. So in this first category, we want to look at those that are non-typical. So specifically, Mycobacterium and Nocardia. They have a cell wall, but their cell wall contains what we call mycolic acid. That's why these two are typically referred to as acid-fast bacteria. And so the mycolic acid component of their cell wall is high in lipid content. And so because it's high in lipid content, these cell walls are resistant to dyes. So if you go to do a gram stain on these two types of bacteria, they will not stain like a normal bacteria would. So because of that, it makes these a little bit harder to identify. It also makes them a little bit harder to treat. The other exception to the rule is a specific type of bacteria called mycoplasmas. These do not have a cell wall at all, and so staining them obviously becomes a problem because the stain is heavily dependent upon the cell wall. So if they don't have a cell wall, they don't stain. It also makes them a little bit more difficult in treatment. We'll talk about why that is a little bit later on. And then, of course, we'll go ahead real quick before we end this segment and look at the cell membrane. Remember, this was the other component of the cell envelope. So we learned in biology that the cell membrane is a phospholipid bilayer. So it's a double layer of phosphates and lipids all bound together. There are proteins embedded in there which do a lot of the functions of the cell membrane. And some of the functions that we see in prokaryotic cell membranes is of course, one thing we talk about in biology, is that it's selectively permeable. So it provides a channel to move materials in and out as it wants to. So some materials are not allowed in and out, which is that selectivity that they have. We just mentioned a channel, so passage of nutrients in and out of the cell is a big function of the cell membrane. But one function that we see for the cell membrane in prokaryotes that we don't see in eukaryotes is, remember, prokaryotes do not have a nucleus. They also don't have membrane-bound organelles. So they don't have mitochondria. They don't have Golgi. They don't have chloroplasts, things like that. And so their cell membrane provides a location for energy reactions, for nutrient processing, for synthesis that would normally have to happen in those organelles. But since they don't have organelles, those reactions are going to happen in the cell membrane. So you remember, here's that phospholipid bilayer, and there's those proteins that are found there. And that's where we'll end part B.